My name is Victor Ramos. I work at the Rockefeller University in the Laboratory of Molecular Immunology. And today we're going to talk about how we are capable to annotate and classify thousands of HIV sequences using our computational pipeline. My presentation is entitled Defective and Intact HIV Genome Assembler, DIHIVA, a pipeline to annotate and classify HIV genomes based on next-generation sequencing data. So recently, several studies have been reporting the HIV genome characterization using either Sanger or next-generation sequencing, but they always implement a non-automated process that relies on manual interventions, which could be really time-consuming due to the high number of sequences the high-throughput technologies are capable to provide. Here, we combine a PCR technique with next-generation DNA sequence based on single genome amplification to characterize the HIV genetic diversity. This technique is called Q4 PCR, and it starts off by extracting the genomic DNA from CD4 cells at limiting dilution, followed by the amplification of near full-length proviral DNA, and the amplicons are then assayed by the Q4 PCR technique that uses a combination of primers and probe sets in 4 different locations of the HIV genome. We select the samples that present at least a double positive signal for any combination of the primers and probe sets. The selected samples are then subjected to a nested PCR and next-generation sequencing. Once we have the FASTQ files, they are ready to be submitted to our pipeline. And here we're going to talk about how we get the classified gene bank sequence from the raw FASTQ files. We start by checking if we have all the files that we need for each sample, and if by any chance they are duplicated. After that, we count how many reads we have for each sample, and if you have less than 500 reads, we classify the sample as empty well. Otherwise, it proceeds to the quality control step that involves PCR amplification removal, correction of putative errors generated during the sequencing reaction, and selection of unique reads. We also perform positive selection using the Los Alamos HIV database, and at this point we filtered out samples that have less than 250 reads, classifying them as empty well. Spades then that implements a diverging graph assembly uses the high quality reads to create a draft of the genome being assembled. And it's required that they have more than 1 KB to proceed, otherwise they are classified as problems in assembly. We blast this draft genome to the Los Alamos HIV database, and by the alignment, we check the orientation of the draft genome in order to export it in the positive orientation. In the step 4, we use our in-house Perl scripts that export a consensus sequence based on the modification of the draft genome by the mapping of the high quality reads to it. We also have another Perl script that export a gene bank file that represents the annotation of the consensus sequence. And at the end of the step 4, we also have the BAM file that represents the mapping of the high quality reads to the consensus sequence. So when we have the annotation and the coordinate of the reads, we can start looking for what we call double peaks. So if a sequence has a double peak inside the M gene or 10 double peaks across the HIV genome, we classify the sequence as a bad assembly. Otherwise, they are classified as a good assembly, and they are subjected to the classification according to the genetic structure. The sequence can also be used for downstream analysis, such as antibody neutralization assays and clonal analysis. And the pipeline finishes its execution by generating an HTML report. All the processes are controlled by a framework constructed in Python called SnakeMake. Here is an example of the HIV sequence classification that we call intact, when we have the 9 genes and the LTR and also the Psi region. We also have the MSD mutation. The only difference from the intact is the mutation in the MSD site. We also have the non-functional sequences that we have early stop codons in at least one of the genes represented by the black rectangles. We also have the misinternal gene sequences that present partial or complete deletion of any gene. We also have the sequence that we call weirds, that are sequences that present duplications or inversions of any gene, and the artifacts, that are sequences that in the extremities we don't have either the PSI or the LTRs. Here's a screenshot of the interface that we built in Shiny in our package, and we can run it using a Docker image to make it really user-friendly. I think that's it. I'd like to thank you, all the authors involved, and you for watching this presentation. Thank you.